When John leaned in, Pedro aggressively lunged at his face with a bite force of 450 pounds. Pedro's lower incisors, which are used for tearing, grabbed John's chin just below his lip. The dog's top left canine tooth dug deeply into John's
Oh no, a dog with rabies just bit me.
close to his eye.
First, a macrophage encounters a pathogen because histamine and prostaglandins are released at the site of the bite. The macrophage engulfs the pathogen and performs antigen processing. This is when a phagosome fuses with a lysosome and breaks up the pathogen into antigens, ready to be presented to a T cell.
The helper T cell then binds to the antigen-presenting cell by means of MHC2 proteins. The binding triggers to the macrophage to release the cytokine interleukin-1, activating T helper cells. Then, the T helper cell releases cytokines stimulating proliferation. Clones of T helper cells are produced with the same T cell receptors. Receptors are all specific for the antigenic determinant of the original processed antigen.
Next is the effector phase, which involves communication between the B and T cells. Activated helper T cells release chemokines that attract B and cytotoxic T cells. B cells arrive at the scene with their IgM receptors. The IgM binds to the antigen, and the cell engulfs both. An enzyme digests antigens into fragments. The fragments are then attached to MHC molecules and are presented on the cell. A T cell receptor binds to a B cell with the antigen being presented. The activated helper T cell releases cytokines that activate the B cell to proliferate.
Next is clonal selection. Resulting B cells then develop into either memory cells or plasma cells. Plasma cells are considered antibody factories, while memory cells are long-living lymphocytes capable of responding to a particular antigen if it is reintroduced.
These antibodies bind to antigens at antigen-antibody complexes. The antibodies inactivate the antigens by neutralization, opsonation, agglutination, precipitation, or complement fixation.
The antigen-antibody complex is called the C1 complex. The C1 complex produces C2A and C4B fragments. The C2A and C4B combine to make C3 convertase. C3 convertase hydrolyzes into C3A and C3B fragments. C3B cleaves C5 into C5A and C5B. C5B, C6, C7, C8, and many C9 proteins come together to form a ring in the cell membrane. This ring allows molecules to pass in and out of the membrane. This causes the membrane to lyse, therefore killing the cell. The man receives a fast-acting rabies shot in order to rid the virus from the body.